actually. Well, we've been hearing about a lot of different applications for kind of substituting what human being already has, as the one that Pedro brilliantly talked previously. Here, I want to talk about what human beings are able to replace, what they can provide as corpse, either living or cadaveric donors, which is the issue that formed the starting point for tissue banking. Tissue banking basically is the possibility of collecting and storing tissues, apart from organs. In this case, I'm talking about tissues that will be afterwards used for transplantation purposes in either allogenic and homologous transplantation. So the idea that is emerging during the recent years is that adipose tissue, which has incredible trophic features that are already exploited into a lot of plastic surgery, and not only plastic surgery applications, can adipose tissue be banked for transplantation purposes? Why do we need to bank it? Why are you thinking about banking it? Because in this case, we will avoid multiple surgeries that are needed to collect adipose tissue to perform several different injections in the host. In this case, we're talking about mainly autologous transplantation, even though given the tolerogenic properties of some of the cells that are included in this tissue may also suggest a possible future application also in allogenic transplantation. So why adipose tissue? Because as you all of us know, it comprises not only completely terminally differentiated cells, which are the adipocytes, but also a trophic stromal vascular portion, which includes stromal stem cells, which are somatic stem cells, similar to the mesenchymal stem cells that are originally described in the bone marrow, but they are not identical, but shares a lot of features. And then also other perivascular cytotypes that are endowed with trophic capacities. There's a universal consensus on the fact that fat is not all the same. We have several different moieties of fat all over the body. We can distinguish white and brown tissue. We can distinguish different depots of white adipose tissue. In fact, it's white adipose tissue, which is the most represented tissue in adult beings, is mostly found in either subcutaneous depots and in visceral depots. And also in this case, the adipose tissue that is found in visceral and subcutaneous tissues depots is not the same. They are characterized by multiple differential features, including biological properties of the stem cells that are included, and also the metabolic-related associated risk. We need to focus on subcutaneous adipose tissue because actually this is the source which is mostly exploited because it is easily harvested through liposuction, lipoaspiration techniques in the procedure that are commonly known as fat grafting procedures. Plastic surgeon has been studying this issue a lot. Today it is like 20 years they've been studying how size and collection procedures may affect the yield of adipose tissue grafting and also the outcome after transplantation. This diagram nicely shows the technical issue, the technical harvesting of adipose tissue through lipoaspiration and what it comes after that. We need to centrifuge this liposucted adipose tissue, and we thus obtain these 3 layers comprising the stromal vascular portion, which is endowed with different cytotypes, as already mentioned, and this is the immunophenotype that is perfectly described. How can we bank the tissue? In this case, adipose tissue. We first need to consider what are we thinking about, or what do we think to bank? Are we thinking about banking the whole tissue, which could be extremely feasible because that means that just right after collection through lipoaspiration, the syringe or whatever vial can be snap frozen in liquid nitrogen or as -80 before. Of course, we will need, as for any other tissue, to cryopreserve the tissue and the cells that comprised inside the tissue. Usually we use the dimethyl sulfoxide as the mostly used cryoprotective agent, even though some toxic issues are still pending. And then we also need to think about how long can we preserve this in this way? Can we bank this tissue? So the main question is it feasible? The feasibility has been already on its way to the confirmation, let's say. This nice work already showed that by electronic microscopy, this is an SEM, that after thawing of cryopreserved fat, you can see that the multilocular architecture of the tissue is still preserved. And in particular, through this other study, they demonstrated that even after many years, up to over 3 years of cryopreservation, the viability of the tissue was completely kept. They observed an expected decrease in cell viability after the first year, but then after the remaining years, up to the third years, the decrease in cell viability was visible. This other study was most focusing on the thawing step. So alternative protocols have been tested, and they realized that the fast thawing, which is actually the procedure that we use when we need to thaw living cells and put them in culture, that means keep them from the nitrogen or from the -80 and kept them at 37 degrees. And that kept completely the viability and cell proliferation capability of cells, and also, all the other biological features including multilineage potential. The final question, which is the one where we actually focus more deeply, is what should we bank? Which depot? Is there any preferential site or any preferential level for liposuction, for liposucting adipose tissue to be banked? This guy, this surgeon, Italian surgeon, many years ago, centuries ago, demonstrated that the subcutaneous tissue is comprised 2 different layers that are separated to the superficial fascial system, which is this white line here. So we have this superficial adipose tissue layer. I will use this SAT acronym during the rest of the presentation. Is separated from this deep adipose tissue layer. Behind this microscopic anatomy, other studies have been demonstrating the anatomical structure of these 2 layers that may act as different tissues, actually, and we demonstrated something more about that. Actually, this is the histological appearance of the 2 layers of the subcutaneous tissue. Here we have the deep layer, and here is the superficial layer. And you can easily realize, even those of you that are not familiar with histology, that these 2 layers are not completely equal. Here we can see a lot of trabeculi that are formed by the superficial fascial system, right below the dermis. This layer of adipose tissue is kind of compressed between the dermis and the superficial fascia, while the deep one is kind of looser without all these retinaculi, and it is also closer to visceral locations, actually. So finally, the aims of this study was in particular to evaluate the differences between this SAT, which is the superficial subcutaneous tissue, and the DAT, aimed at identifying biological features that may help us prioritizing the site for collection and biobanking hence. So the first set of our study design was based on the recruitment of female patients with homogeneous distribution for BMI and absence of associated morbidities and no smokers. And we collected from each patient 5 ml of liposucted fat from superficial and deep location in the subcutaneous tissue from these 3 regions, abdomen, thigh, and knee. Afterwards, we first analyzed the gross anatomy and the histological aspect of this liposucted tissue by by standard histology, and then we process the lipoaspirate to obtain the stromal vascular fraction, so isolate cells, characterize cells, put them in culture, and observe the biological features, including multilineage potential and gene expression analysis, gene expression profiles. This is not going, okay. This is the histological view. Not much to say. The integrity of the tissue can be maintained both using the standard so-called Coleman technique, which use a larger cannula for lipoaspiration, and using the micro cannula that we need to use to separate to obtain selected superficial or deep harvesting. And this is confirmed using histological staining, which in this case, hematocylin eosin staining, allow us to confirm that also the stromal population are preserved, which is important because there all the stem cell resides. We also demonstrated that within the stroma we have cells that are capable of proliferating. Means that here we found the adipose stem cells that are kept, adipose-derived stem cells that are kept in the lipoaspirate, as we demonstrated by using the Ki67 staining. And then we analyze what happens upon fractionation of the tissue through centrifugation, which is the very first step. We obtain the usual 3 layers: oil layer, tissue layer, and blood saline layer. This is, of course, just a representative image. We repeated that for 13 different patients, and we saw that the oil fraction, the volume of the oil fraction, normalized for the volume of total lipoaspirate, was always higher in deep harvest compared to superficial one, meaning that we have more traumatism to the during the harvesting procedure. We have increased volume of the tissue portion, which is the one that we need actually also afterwards for banking and transplantation, which is always increased in superficial harvesting, in particular in these 2 regions, while as you can see the knee also for the blood saline portion doesn't show statistically significant differences. So we can see that the tissue fraction, the volume of tissue fraction, this layer here, is always increased in samples obtained from the superficial side. And then the blood saline fraction is increased in the deep abdominal harvesting and is decreased in thick, while no significant differences are found in the knee. Most differences that we observe between superficial and deep adipose tissue were observed in the abdominal region, where actually the depot is larger. I mean, it depends, of course, on individuals, but it is usually larger, while in the knee we have something that is more like a fat pad, which retains homogeneous characteristic throughout the deepness of the thickness of the depot. Then we counted viable cells. Viable cells were actually higher, significantly higher in superficial harvesting in both the abdominal and the thick region, while again in the knee we didn't see any statistically significant difference. And then we tried to analyze the number of cell count regardless of the region of collection. So again, set is higher, display higher viability compared to that, to the AT. And when we exclude the knee, sorry, the animation was kind of confused. When we exclude the knee, we have even an increased statistical significance, so decrease in the p-value. Then we assess the immunophenotype. All these cells, of course, most of these cells display the immunophenotype of this heterogeneous population, which represents the SVF. In particular, we saw that we have a statistically significant different number of CD105 positive cells, meaning stromal cells, perivascular stromal cells, which are basically, at least in part, overlapping with the so-called adipose-derived stem cells in the superficial adipose tissue compared to the deep adipose tissue, while in the others we didn't observe any significant difference for the other markers. No significant differences in morphology and also in cell growth kinetics, so the cells we were able to isolate by plastic adherence in culture were all the same, which is expected because what we can select by plastic adherence is a more homogeneous population of cells, of course. And then we wanted to, then we wanted, this is not working properly, and I apologize for that. Anyway, this is it is disappearing, but what I wanted to show is that we observed the increased expression of markers that are related to stemness potential of cells, in particular NA-NOG gene, POF gene, PO, UF, and SOX2. These are embryonic stem cells markers which are believed to be related to the stemness potential, multilineage potential, plasticity of cells, and possibility of stem cell self-renewal. So these marker genes were significantly upregulated in cells isolated from the superficial adipose tissue compared to deep adipose tissue. And then we also demonstrated that, of course, cells from both sides are able to differentiate, but those that are isolated from superficial tissue display an increased tendency to in vitro differentiation in both adipogenic and osteogenic lineage. In particular, those that were isolated from this SAT, from the superficial side, were able to form in vitro sizable and microscopically evident spheroid aggregates that were stained with alizarin instead. This was staining. This is what occurred upon osteogenic differentiation induction, and this is what occurred upon adipogenic differentiation in vitro. So a bone nodule, an initial bone nodule, and an adipogenic nodule with oily droplet inside the cells. So the gene expression profiling of performed during during induction of differentiation, so during the differentiation assay in vitro, confirmed the increased differentiative potential in vitro of cells isolated from the SAT. And then we started, these data are completely preliminary and were performed on a reduced sized sample size. So I just need to confirm this in further patients, of course. But we observed that the differences between set and DAT in terms of expression of this marker, stemness marker, were kind of age-related, because the differences were more evident in aged person compared to the group that were below age 44, because these were the two cohorts that were available actually. So that means that when we are younger, we don't have as many differences in the two regions, but during aging, the differences increase, meaning that the content in stem cells in the deep subcutaneous tissue tend to decrease with age, and also they tend to decrease with BMI, tend to increase with BMI. But still, these were completely preliminary, so we want to confirm this anyway. Part of these results have been already published on cytotherapy recently, while the second part of this study is completely preliminary. So I will be extremely rapid, because we wanted to assess whether the possibility of biobanking superficial or deep adipose tissue displays some differences. So we did independent experiments on another set of patients with the same inclusion criteria. So we collected samples and we cryopreserved portion of the sample of the entire adipose tissue and used fresh adipose tissue of the same donor as control. So we counted viable cells. The number of, these diagrams are still in Italian, I can easily translate. These are the number of cells, or total cells in this case, viable cells in this case, and the dark red is superficial, while the light red is deep, as you can see. So the differences between superficial and deep is significant, as we already demonstrated, so this is a confirmation, but the differences increases after thawing, meaning that cells containing the SVF of the superficial adipose tissue are more resistant, let's say, to the traumatic injury that occur during freeze and thaw procedures. Also, the mortality after freeze thawing is kind of increased, indeed is lower in cells obtained from superficial layers compared to the deep one. And this is just an overall, just to give you an overall idea of which are the number of viable cells across all regions. This is abdomen superficial, abdomen deep, thick superficial and deep, and then knee superficial and deep. And this is what occurs in this set of samples actually, in fresh sample. And this is what happens after thawing. So superficial is still higher. So we still have more viable cells in superficial tissue after thawing than those that we can observe after freezing and thawing. Of course, all thaw cells maintain their in vitro multilineage potential. So the conclusion our conclusion are that superficial and deep subcutaneous adipose tissue are different, not only for the location, but for the biological properties and for the properties of the stem cells that are included in their stroma. These differences are basically region dependent because we can see those differences mainly in the abdomen, less in the knee, for instance. We didn't try all the region of the body, of course, and we believe that may have also this age and BMI effect that we need to confirm, of course. So can we say that the superficial adipose tissue is a different kind of tissue? It's not a matter of level, it's a matter of tissue. It's kind of a different moiety of adipose tissue, and though with a higher plasticity and a regenerative potential based on the stemness features of this portion. Thus, this may suggest some kind of a prioritization of the level for liposuctions, for plastic surgeon, or any other application for either surgery, but also for biobanking procedures. And I wish to thank all the persons that are involved. The red underline are the young that mostly perform all the work. And I wish to thank also you for the attention and all the granting institution. Thank you.